In this video, we will be explaining the central dogma of biology, which states that DNA makes RNA and RNA makes protein.
Replication is a process in which a DNA molecule is copied to make two identical DNA molecules. Replication begins when the enzyme helicase unzips the double-stranded DNA into two single strands of DNA, one being a leading strand and the other a lagging strand. The two separated strands of DNA create a Y-shaped space called the replication fork. The leading strand goes in a 3 to 5 direction, whereas the lagging goes in the opposite direction, which is 5 to 3. After the strands are separated, an enzyme called primase places a segment of RNA called a primer, which acts as a starting point for making DNA. An enzyme called DNA polymerase connects to the leading strand from the primer and goes along the entire strand, adding corresponding nucleotides in a 5 to 3 direction. For the lagging strand, the primase places a primer to make a starting point for the DNA polymerase. Since DNA polymerase only goes in a 5 to 3 direction, on the lagging strand, the DNA polymerase adds little segments of nucleotides called okazagi, and the rest are filled with primers along the strand. After both strands are full, a molecule called exonuclease comes in and removes all the RNA primers, and DNA polymerase fills in the rest of the gaps. So the entire strand is just DNA. After all of it is DNA, an enzyme called DNA ligase scans over both the strands of DNA to seal the nucleotides together.
After replication, transcription occurs, which is when the DNA template is utilized for mRNA synthesis. This process occurs in 3 stages: initiation, elongation, and termination. In initiation, the DNA molecule separates in order for the RNA polymerase to bind to the Tata box. The RNA polymerase has a high affinity for the region sequences of T's and A's. Next is elongation, where the RNA polymerase moves along the template strand and starts transcribing by the new mRNA by adding base pairs that are complementary to the DNA template strand. For example, A pairs with U, T pairs with A, G pairs with C, and C pairs with G. Note that the RNA transcript should be identical to the DNA coding strand except with U's in place of T's. The RNA polymerase continues to transcribe along the template strand until it finally encounters a stop signal, in which it has then reached the stage of termination. The new mRNA is ready to be translated.
Translation is the process in which a protein is made from information contained in a molecule of messenger RNA. Translation occurs in a protein called a ribosome. Ribosomes have a small and large subunit and is a complex molecule made of RNA molecules and proteins. Translation of an mRNA molecule occurs in 3 stages that are called initiation, elongation, and termination. During initiation, the small ribosomal subunit binds to the start of the mRNA sequence. Then, the large ribosomal subunit binds to form the complete initiation complex. During the elongation stage, the ribosome continues to translate each codon in turn. Each corresponding amino acid is added to the growing chain and linked by a bond called a peptide bond. Lastly, termination occurs when the ribosome reaches a stop codon, UAA, UAG, and UGA. Since there are no tRNA molecules that can recognize these codons, the ribosome recognizes that translation is complete. The new protein is then released and the translation complex comes apart.